Imagine you were given basically an impossible task. You need to map every single road, every dirt path, and like every hidden alleyway across an entire continent.
Right, which alone would take lifetimes.
Exactly. But the task doesn't stop there. You also need to know the exact occupation, the personality, and, the daily schedule of every single person living in the houses at the exact moment you draw the map.
Yeah, you aren't just mapping the geography. You're trying to map the living, breathing identity of the entire space simultaneously.
And just thinking about the sheer scale of that kind of data collection is honestly paralyzing.
Oh, absolutely. Historically in biology, you kind of had to choose.
Right.
You either mapped the physical wiring drawing the roads, or you took a cellular census to understand what the cells were doing. Doing both at the microscopic scale of synapses seemed, well, fundamentally incompatible.
And that right there is exactly the bottleneck scientists face when trying to map the mammalian brain. So in today's deep dive, we are exploring this fascinating 2026 paper from Nature Methods.
The Connectome-seq paper.
Yeah, Connectome-seq. It introduces a truly groundbreaking technology, and our mission today is to really understand how scientists have invented a way to map the brain's wiring diagram, the connectome, while simultaneously reading the genetic mail of the connected cells.
And doing it all using customized viruses and molecular barcoding, which is just brilliant.
It really is. So for you listening, if you've ever wondered how your memories are physically stored or, why brain diseases like Alzheimer's are so infuriatingly hard to untangle, it's because we haven't had a proper map. And you are about to learn how that is fundamentally changing.
To really grasp why this is such a paradigm shift, we have to look at where we are coming from. We actually have mapped entire brains before.
Wait, really? Entire brains?
Yeah. We drew the complete wiring diagram for roundworm C. elegans, which has about 300 neurons.
Okay, 300 is manageable.
Right. And recently, scientists pulled off a massive feat and mapped the brain of a fruit fly, Drosophila, which is around 140,000 neurons.
Wow.
But the method used there involves taking the brain, slicing it into unimaginably thin sections, photographing every single slice with an electron microscope, and then using computers to stack those images back together.
Just to physically trace the wires?
Exactly.
But mammalian brains are a completely different universe of complexity, right? A mouse brain has roughly 70 million neurons.
And a human brain has 86 billion.
86 billion. You can't just scale up that slice and photograph method for a mammalian brain, especially if you want to study how brains differ from, individual to individual.
No, you really can't. It takes years and massive computational power just to map a volume of tissue the size of a grain of sand using that old method.
Which leaves us looking for biological shortcuts. We need something high throughput.
Yeah.
But wait, haven't scientists been using viruses for years to trace brain wiring?
They have, yeah.
Because I've read about researchers injecting rabies viruses into the brain because the virus naturally jumps from one neuron to the next across the synapse. So why not just use those to draw the map?
Well, those viruses are essentially ticking time bombs.
Oh, really?
Yeah, traditional viral tracers like rabies are highly toxic to the cells they infect. They jump across the synapse, sure, but they eventually kill the neuron.
Ah, I see.
That severely restricts your experimental time window. You only have a few days before the circuit you are trying to study is completely destroyed by your own tool.
That's a huge problem.
And furthermore, in the densely packed mammalian brain, where millions of connections overlap like the world's worst spaghetti junction, the signals from those viruses get all mixed up.
So you lose the precise resolution.
Exactly. You lose that single-cell resolution. We needed a tool that doesn't just safely show that two specific neurons connect, but tells us the exact molecular identity of those neurons.
Okay, let's unpack this. Because the researchers developed Connectome-seq to solve this exact problem.
They did.
And to understand how it works, we need to look at the elegant molecular engineering involved. I like to think of it like outfitting neurons with a two-part molecular Velcro.
That's a great analogy.
Where each side of the Velcro also carries a unique shebang manifest. So they start by using AAVs, adeno-associated viruses, which are incredibly safe and minimally toxic.
Yes, AAVs are sort of the workhorse of safe gene delivery right now.
So they use these as delivery vehicles. They inject these viruses into the brain to deliver specific genetic instructions to the neurons, basically telling them to build a specialized artificial protein anchor.
Right, what they call the SynBar system.
The SynBar system, exactly.
The architecture of this SynBar system is just brilliant. Every connection between neurons has a sending side and a receiving side, separated by a microscopic gap called the synaptic cleft.
Right.
So the viruses force the sending neuron, to build one half of the Velcro. Let's call it the pre-protein. And this is actually built from a natural protein called Nerexin-1 beta.
Okay, Nerexin-1 beta for the pre-protein.
Yes. And they force the receiving neuron to build the other half, the post-protein, which is built from neurologen-1. These aren't just random proteins. They inherently want to bind to each other.
So when they meet across that tiny gap, they just lock together tightly.
Exactly.
But a locked connection is completely invisible unless you can actually see it. So they engineered a glow-in-the-dark feature, which I think is so cool.
That's a split GFP. It's so clever.
Yeah. They took a green fluorescent protein, GFP, cut it exactly in half, and attached one half to the pre-protein and the other half to the post-protein.
Right. So when those two sides of the Velcro meet and lock across the synapse, the two halves of the glowing protein snap together.
And that specific connection literally lights up green.
It does. But what's fascinating here is the sheer amount of trial and error required to make the next part work.
The shipping manifest part.
Exactly. Lighting up the synapse is great, but it doesn't tell us the identity of the cells. So along with the Velcro, the viruses also instruct the neurons to manufacture unique RNA barcodes.
Okay, so the sending neuron makes a unique pre-barcode, and the receiving neuron makes a unique post-barcode.
Right.
And these barcodes are massive random sequences of genetic letters. I was looking at the math here, and it is wild.
The 30 nucleotide random sequences.
Yeah, because the sequences are randomly generated from 30 genetic letters, it creates so many billions of possible combinations that statistically, every single neuron in the brain gets a completely unique, unmistakable barcode.
Which is perfect for identification. But an RNA barcode floating freely inside a cell does us no good.
Right, it has to be at the synapse.
Exactly. We need those barcodes to be physically held at the synapse, right where the connection happens. To do this, the scientists engineered a specific molecular hand onto both the pre-protein and the post-protein.
And this hand is called the lambda N22 domain, right?
Yes, the lambda N22 domain. It's designed to grab a specific sequence on the RNA barcode and hold it tight. But, and this is where the engineer gets crazy, when they first tried putting this molecular hand at the very end of the Velcro protein, it completely ruined the protein's shape.
Oh, so the Velcro wouldn't stick anymore.
Exactly. Just broke the locking mechanism. So they had to go back to the drawing board and molecularly hide this hand internally within the protein structure. Yeah, so it could hold the barcode without interfering with the locking mechanism.
That is incredible. But wait, there was a massive physical transport problem to solve too, right? Because a neuron isn't just a simple sphere.
Not at all.
The receiving neuron builds its barcode near the main cell body, which is relatively close to the synapse. But the sending neuron builds its barcode in its own cell body, which could be located on the entirely opposite side of the brain.
Right, down a massive, long biological cable called the axon.
So how does the barcode get all the way down there?
Well, left to its own devices, that genetic barcode would never make the journey. It would just sit in the cell body.
So they had to attach an engine to it.
Precisely. They combed through the genetic literature, testing different natural transport sequences, until they found one, the 5' UTR of a gene called RPL7.
RPL7. Okay, so this acts like an active zip code.
Yes. When attached to the barcode, this RPL7 sequence actively hijacks the cell's internal transport highway, dragging the barcode all the way down that long axon to the distant synapse.
Okay, I want to do a quick recap here to lock in a mental picture of what we have so far, because there are a lot of moving parts.
Good idea.
So step 1, we've injected safe AAV viruses into a living brain. Step 2, those viruses have instructed the neurons to build molecular Velcro that reaches across the gaps between cells and locks together, glowing green.
Right.
And step 3, each side of that Velcro is holding tightly to a unique genetic barcode that identifies exactly which cell it belongs to, thanks to that internal molecular hand and the RPL7 transport engine.
Spot on. You have successfully tagged millions of specific microscopic connections in a living, breathing animal.
That map is completely useless if it stays locked inside the skull.
Exactly.
I mean, how on earth do you extract these microscopic fragile connections without breaking them apart?
The physical extraction process is arguably as impressive as the genetic engineering. And it brings us to the concept of the synaptosome.
Right, because it's one thing to tag the synapses, but gathering them, the researchers essentially put the brain tissue into a blender.
Whoa, very, very gentle one.
Right. They use a specialized sugar solution that cushions the cells and perform gentle homogenization, which honestly sounds like the ultimate scientific euphemism for putting a brain in a NutriBullet.
It does sound violent, but the fluid mechanics of this sugar solution are highly controlled. When you apply this gentle blending force, the long, delicate biological cables, the axons and dendrites, are sheared off.
Okay.
But the synapses themselves, where our molecular Velcro is locked tight, are structurally very dense. When the cables shear away, the broken cell membranes surrounding the synapse instantly snap shut and reseal themselves.
I like to picture a long, thin balloon. If you twist it and pinch it off in the middle, it forms a tiny self-contained bubble.
That is exactly what the cell membrane does. It snaps and instantly reseals into a tiny, secure escape pod called a synaptosome.
An escape pod, I love that.
And these microscopic floating orbs contain both the sending and receiving sides of the synapse locked together, with their unique RNA barcodes safely trapped inside.
And simultaneously, this blending process also releases the intact nuclei of the neurons, right?
Yes. The nucleus is the control center. It contains the main blueprint of the cell, the transcriptome, which tells us exactly what kind of cell it is, along with a master copy of its unique RNA barcode.
So now we have a test tube filled with this cellular soup. It's a mix of free-floating nuclei and millions of these tiny synaptosome escape pods.
Right, and we need to fish them out.
Which presents a massive sorting challenge. They use a flow cytometry machine, or FACS, that fires lasers at the soup to sort the structures based on their glowing colors. But immediately they hit a spectral wall.
They really did, because they needed to isolate single nuclei. And to confirm they had a nucleus, they needed to use a chemical dye that binds to DNA.
Okay, makes sense.
The problem was that the standard DNA dyes scientists use, like DAPI, well, their visual signals overlap with the green of our engineered Velcro.
Oh no, so the lasers in the sorting machine couldn't tell them apart.
Exactly.
It's like trying to find one specific person in a massive crowd who is wearing a green shirt, but you accidentally forced everyone else in the crowd to wear green shirts too. It's a visual nightmare.
That's a perfect way to put it. To bypass this, they had to pivot and use a highly specialized far-red dye called SIR-DNA.
SIR-DNA.
Right. This shifted the DNA's glowing signature into a completely different invisible part of the color spectrum. The machine's lasers could now easily distinguish the green glowing Velcro from the far-red glowing DNA.
Brilliant. But the tiny synaptosome escape pods were even trickier, weren't they?
Oh, yeah.
That split green glowing protein we talked about, the split GFP, it turns out inside this high-speed laser sorting machine, that reconstituted glow just wasn't bright enough to be detected reliably.
No, they were basically invisible to the machine. The GFP is great for looking through a microscope, but not for high-speed laser sorting.
So what did they do?
They engineered tiny molecular flags onto the outside of the Velcro proteins, specifically V5 and HA tags.
Okay.
They then flooded the soup with highly fluorescent antibodies. These are immune proteins specifically designed to seek out and attach to those tiny V5 and HA flags.
So once those glowing antibodies latched on, the synaptosomes finally glowed brightly enough to be plucked out of the cellular soup.
Exactly.
Okay, so we've gently blended the brain, created our escape pods, and used lasers to sort out our perfectly intact tagged synapses and our cell nuclei.
We have our isolated data points.
Now for the real magic, reading the data. And for this, they use advanced 10x genomics microfluidic chips.
Right.
So picture this. A tiny glass slide with millions of microscopic channels. Through precise fluid dynamics, one single nucleus, or one single synaptosome, gets trapped inside a tiny droplet of oil.
And inside that oil droplet with it is a single microscopic gel bead.
Right. Tell us about this gel bead.
That gel bead is a marvel. Imagine a tiny microscopic planet, and growing out of this planet are 3 different kinds of chemical trees.
Okay, 3 trees.
Each tree is designed to catch a specific type of genetic leaf. One catches the cell's standard messenger RNA to identify the cell type. The second tree is designed to exclusively catch the pre-barcode. And the third tree catches the post-barcode.
And because they are all captured simultaneously inside that isolated oil droplet, there is no mixing of signals, no competition.
None at all.
The sequencing machine reads the exact genetic identity of the cell and its unique shipping manifests all at once.
All at once. Let's anchor what we've just covered before moving to the punchline, because the application of this is wild.
Yes, let's do another quick summary.
We've gone from intact brain tissue to isolated data points. We used a cushion blender to force the connections to pinch off into secure escape pods.
Yep.
We used far red dyes and glowing immune proteins to sort those pods with lasers. And finally, we trapped them in tiny oil droplets with specialized beads to read their genetic mail.
It's like a sci-fi assembly line.
It really is.
Yeah.
But the ultimate question is, what happens when you take this incredibly precise molecular sorting machine and unleash it on a real breathing neural circuit.
And here's where it gets really interesting. They road tested this entire Connectome-seq system on a mouse. Specifically, they looked at the pontocerebellar circuit,
Right.
which is this massive long-distance neural highway connecting the pons, which sits deep in the brainstem, to the cerebellum at the back of the brain.
And this is a classic, heavily studied circuit. For decades, the biological consensus was completely clear. Fibers from the pons project up into the cerebellum and connect to very specific targets, like granule cells.
And Connectome-seq confirmed this instantly, right? The data spit out thousands of these expected textbook connections.
It did, but the data also spit out something that made the researchers do a double take.
Yeah, they found a robust, undeniable set of connections going directly from the pons to a completely different type of cell called a Purkinje cell.
Right.
Now, if you open literally any neuroscience textbook printed in the last 50 years, it will tell you unequivocally that this specific connection is purely transient.
Transient, meaning it exists for a brief window when a mammal's brain is developing and then it disappears completely in adulthood.
Exactly. But they found it in adult mice.
And discovering an impossible connection in an adult brain is heavily scrutinized. Some scientists had actually seen fleeting anatomical hints of it before, but without genetic proof, those hints were largely dismissed as experimental errors.
Just background noise.
Exactly. Connectome-seq changed the game because it didn't just show two cells touching, it provided the definitive transcriptomic proof.
Because the system reads the entire genetic expression of the cells alongside the barcodes, the researchers could tell the computer to mathematically isolate the exact Purkinje cells that were connected to the pons.
Right, they grouped those specific cells together and looked deeply at their genetic blueprints.
And what did they find?
The blueprint was stunning. These specific Purkinje cells weren't just random outliers, they had a wholly unique molecular signature.
Wow.
The data showed a massive enrichment of specific genes related to learning and memory. For instance, they found high levels of a gene called GRID2IP.
GRID2IP.
Yeah, GRID2IP. It's heavily involved in synaptic organization, essentially the scaffolding that holds the connection together.
Okay, so it's a structural gene.
Right. They also found heightened expression of CAC-NAGE
And what does that do?
that controls ion channels, which dictate how excitable the neuron is.
So they literally discovered a genetically distinct subpopulation of cells dedicated entirely to a connection that wasn't supposed to exist.
Exactly.
This isn't just a misprint on the old map. It's like discovering a completely unmapped, thriving neighborhood in a city you thought you knew perfectly, complete with its own unique cellular culture.
It's exactly like that. But, this raises an important question, though. The technology is dazzling, but is it flawless?
Right.
As with any pioneering method, we have to look critically at the limitations of Connectome-seq version 1.0.
Absolutely. It's not perfect yet. Let's talk about the hurdles. First, the matching efficiency is currently hovering around 12%, right?
Yes.
So that means out of all the matched populations they process, they can confidently validate only about 12% of the connections.
And that lower efficiency comes down to the brutal reality of molecular biology. When you extract these barcodes, some inevitably get damaged.
Sure, they're fragile.
Furthermore, to read the DNA, you have to multiply it through a chemical process called PCR. That multiplication process occasionally introduces chemical typos into the barcodes.
Oh, I see. And to fix those typos, they use an algorithm that basically acts like a molecular spell checker, right?
Right.
If a barcode they read is only a few genetic letters off from a sequence they know exists, the computer assumes it was a multiplication typo and autocorrects it to match.
Precisely. It corrects the errors to save the data, but it's a tightrope walk.
How so?
Well, if they make the spell checker too loose, it will start matching barcodes that don't actually belong together, creating fake biological connections.
Oh, that would be bad.
Very bad. So they have to keep the algorithm very strict, which unfortunately means throwing out a lot of potentially valid data just to be safe. Hence the 12% efficiency.
Got it. There's also the observer effect to consider, right? The very act of measuring something might change the thing you are measuring.
Yes, and this is crucial. Remember that our molecular Velcro, those modified neurexin and neurologen proteins, are naturally occurring synaptogenic proteins.
And synaptogenic means they naturally signal the brain to create synapses.
Exactly. By using viruses to force neurons to express high levels of these Velcro proteins, there's a real risk we might accidentally encourage the brain to build artificial bridges that wouldn't normally exist.
It's like trying to map the existing roads in a forest, but doing it by dropping tons of fresh asphalt everywhere you go. You might accidentally build a new road just by being there.
That's a fantastic analogy. The researchers mitigated this by keeping the viral expression window incredibly short, just 2 to 3 weeks.
That's enough time to tag the existing connections, but generally not enough time for the brain to completely rewire itself with fake synapses.
Right. Still, it is a biological caveat they have to monitor. Additionally, right now, this system only works well for long-distance circuits.
Why is that?
Because you need to inject 2 different viruses, one for the pre-Velcro and one for the post-Velcro. You need 2 physically separated injection sites in the brain to prevent them from mixing too early.
Oh, I see. So you couldn't use this current version to map a tiny, dense local circuit where all the cells are completely jumbled together.
Exactly.
But even with those version 1.0 limitations, being able to capture the molecular identity of connected neurons at a single synapse scale is just a monumental leap forward.
It really is.
We've taken an incredible journey today. We started with the paralyzing biological bottleneck of tracking dense mammalian neural wiring. We explored how scientists engineered molecular Velcro and unique RNA shipping manifests.
Yeah, the SynBar system.
We saw how they used cushioned blenders and tiny oil droplets to extract that genetic data, ultimately resulting in the rewriting of a cerebellum neural pathway that textbooks insisted was impossible.
If we connect this to the bigger picture, the integration of physical wiring with genetic identity is what makes this so revolutionary. We are no longer just looking at a static map of roads. We are looking at the traffic, the cargo, and the destinations simultaneously.
So what does this all mean for you, the listener? It means we are entering an era where brain mapping isn't just about drawing cold, static lines between regions. It is about understanding the active genetic conversations happening at individual microscopic junctions.
Exactly.
Think about complex psychiatric or neurological conditions like schizophrenia or autism, where the brain's physical structure often looks completely normal on an MRI, but the communication is altered.
Right, it's a software problem, not a hardware problem.
This technology could completely change how we target drugs. We won't just be blindly treating a general brain region with a sledgehammer. We could eventually target specific genetic conversations at the exact specific synapses where the miscommunication is happening.
And, I'd like to leave you with a final thought to mull over.
Please do.
We know that mammalian neural wiring is highly unique to us as individuals, shaped by every experience we have.
Right.
If our wiring is that personal, and we now possess the technology to read the precise genetic state of a single specific memory pathway down to the individual synapse, what does that mean for the nature of human identity?
Oh, wow.
Are our quirks, our deepest habits, our fears, and our most cherished memories eventually just going to be fully readable as a massive sequence of paired RNA barcodes locked together in microscopic droplets of oil?
That is a staggering thought to process. It really makes you look at your own memories a little differently.
It certainly does.
Well, plenty to think about. Until next time, keep diving deep.